I'll make a note to make sure that's good.
Yeah. Hopefully time. I mean, we're supposed to have the final protocols in August, but I mean, people are late. When I have like 67 people actually like working at the same time, and people have all kinds of delays, and so I mean, in the end, it might be a little later. I'm not sure. Like our original deadline was like early August for having set protocols, so like we'd be including them like between August and September. Time. But it could take a little longer. So Lenny is.
Let me check if Lenny can join. Oh, he just asked us, is Zoom working for me? Let me see if I can send him the Zoom.
Cool.
Oh, but I think the Zoom, when I registered, it gives me like I'll just send him the registration thing.
Can we use the same link for both accessing, Juliana?
No, he receives them.
Can you send one to Lenny or?
Yes,
or
I send one for him, but I can send again.
Yeah, we could also send a general one, right? And
okay.
Oh, okay.
But yeah, just resend it if it doesn't.
I think I got one that was like super long, and so I thought it was like my own.
Yeah, I think that was your own, so I'm not sure you can.
I sent yes, I sent again.
Somebody else. So yeah, there he is.
Okay, there he is.
There he is.
Hello, we'll see you.
That worked.
Hi.
Hello.
So I guess we're 4 minutes away from the official time, so we might as well wait a bit.
Hi, Olavo.
Hi, how are you doing?
Excellent. And Anita, you and I will be running from the same Google Doc. So we'll just take turns who's doing screen share. And if it's a problem, then maybe you'll run it. And because you have more of the demo part, then you'll run it. And I'll just ask you to advance the slides if we're having trouble passing it back and forth.
I think it should work.
It should, but just in case.
Yeah, well, do your thing. If you want help, Juliana is good at managing this kind of thing, but I think you're probably used to it.
Yes.
Excellent. The agenda looks good, right? Hi, Juliana.
Hi. If you want to test, you can share your screen. Above, I have some place in green, and you can share. After, if you want change, you stop, and Lenny can share his.
Or if you want to do like the one share and both speak, I mean.
Yes.
It's up to you. I mean, we'll just wait for everybody to. So who's sharing now?
Yes, you share.
Yeah, that's fine. Okay, I guess we'll just wait 3 more minutes and chat a bit. So we have some people who are actually participating in the initiative, I guess, as watching. I recognize there's like 2 or 3 names. So, I mean, feel free. I mean, those guys are very, I mean, we've talked to them before. They are very, very helpful. So, I mean, be sure to ask and provide feedback. I mean, this is actually an opportunity for us to actually figure out how to best use their platform in terms of actually working within the initiative, besides sharing the protocols, etc., which we'll naturally do. So we'll just wait for a couple more minutes. Welcome everybody who's joining.
Olavo, você que vai compartilhar então a tela?
Eu não vou compartilhar nada aqui e tal, só quem compartilha vão ser eles.
Ah, tá bom. Quem está compartilhando é a Anita?
É um dos dois ali e tal.
Ah, ok. É porque tá pequeno, só pedir para eles se eles puderem colocar.
Can you go full screen? Let's just put, I mean, in Google Docs. I mean, because you see the whole Google Docs.
Yes, thank you.
Yeah, that looks better.
Lenny, I think you're muted. It's Lenny talking.
Anita, can you try to wrestle away control from me and you share screen? Will it prompt me to stop sharing, or do I need to stop before?
No, I can start sharing too. So
Okay,
But you need to stop, don't you?
I can do it, but it says if I share, then yours will go away, but I can do it. So
okay.
excellent.
Yeah, it's now I see something else that is quite confusing.
Yeah.
Yeah, somebody should stop before.
Yeah.
Okay, excellent.
Okay, so whoever's starting can. Well,
So you give us the signal, Olavo, and
we're on minute. We're 1 minute. Oh yeah, so this is time. So I guess yeah, we might as well start. So well, welcome everybody who's joining either via Zoom or via Facebook. We're the Brazilian Reproducibility Initiative. We have this monthly webinars, as you probably don't know at this time. So our guests today are Lenny Teytelman and Anita Bröllochs from protocols.io. We've been speaking to them for a while. So I met Lenny at the Force 11 meeting last year, which is a meeting on scientific publication and etc. So that's when he actually gave a very nice talk on the history of protocols.io and just like how hard it is to make something like this work. But that was more or less at the time we were developing the whole protocol for the initiative, and naturally we want to share our protocols in the. I mean, we're doing a lot of work on protocols at the moment, as you who are in the initiative probably know. I mean, we're going to be working on this for like a month or two, so we'd be very happy to actually put this in a format where we can share this more usefully with people once they're ready. Of course, that's like a long time away. That's like after the experiments. But even beforehand, I mean, Protocols is an interesting platform in terms of like managing protocols and actually executing experiments. So I guess Anita and Lenny will be able to show us a little bit more of how it works, and we can all have a discussion on how to best use this within the initiative. And I really appreciate everybody's ideas on this. So I guess you can go on. Whoever's starting. Go.
Thank you, Olavo. Thank you for the invitation. It's an absolute pleasure. Before Anita and I start, just a disclaimer, I am technically officially on vacation starting an hour ago. And so I'm in a house, it's a family vacation. I'm in a house with 5 really little kids running around, and at any point the door might open and you might see a monkey peeking in. If that happens, I apologize. But otherwise, I think this should be a really fun conversation. We'll have 2 parts. In the first part, we'll go over just not the history behind Protocols.io, but the idea of what our mission is and what is the general issue, which I'm sure for those who are in the Reproducibility Initiative, as Olavo said, is already very familiar, but what's the general issue of protocol reporting? We do have materials and methods sections in paper. So I'll talk a little bit about that. And then Anita will very quickly, in like 10 minutes, go over some of the key features of the Protocols.io platform particularly, and then I'll fill in a couple of minutes where we are now, what the adoption is like. But the most important part for Anita and I is the Q&A in that first part. So any questions, requests for what you'd like to see that's not there, questions about maintenance, business model, long-term preservation plans, we really are looking forward to the Q&A part of this. And then in the second part, the second half hour, we'll talk less about the platform itself and more about what are the best ways of sharing methods and details, because platform is just a vehicle for sharing the knowledge, but how you share it, how you structure it, requires some thought. So we'll go through that in part 2, and then we'll also have Q&A. So with that, I mentioned method sections of papers, and just to set up what the mission of Protocols.io is, as far as the introduction, here is a biologist who said on social media, I'm looking for a paper in 97 paper, as described in 96, finds 96 paper, as described in 87, finds 87 paper, it's too old, library doesn't have subscriptions, it's paywalled, which is a very familiar and frustrating experience to anybody who's come across a method section like that. And this is not just in biology, of course. Here is a physicist who says, reading through methods, devices fabricated as previously described, previously described, previously described. The original reference says devices were fabricated with conventional methods, right? And this, of course, is really the state of the art of many, if not most, of the published research papers. We theoretically have the space in the materials and methods to describe exactly what we've done, but in practice, that space is often constrained. There may be constrained, there are page limits, and we just don't have the culture of really sharing the detailed step-by-step protocols. So we end up with things like this, where you can get the gist of what was done, you know which methods were used, right? They extracted DNA, then they used the following programs to analyze, but what were the parameters of the programs that were run? What were the details of the DNA extraction? That is often missing. So the mission of Protocols.io is to improve particularly this part, both before we're publishing, to make it easy to share the details of what exactly we're doing as you're developing methods, make it easier during publication to include links to the recipes exactly with the details that are necessary for someone else to reproduce what was done, and having a central place where after publication, you can easily share corrections and optimizations of what was done, because methods are not static. They're not just a figure in a paper. They evolve, right? By the time you've published a paper, you might know that 30 degrees works better than room temperature in a particular step. And so having a place where long after you've published, you can come back and share how you've optimized or corrected certain steps is very important. So that's the mission and what we think about daily at Protocols.io. And with that, I'll hand it over to Anita to quickly go over some of the main things that we've developed since we launched in 2014.
All right. So the main functionalities on Protocols.io, I'm just going to go really quickly through it. And so one thing I want to start with is the protocols on Protocols.io are not static PDFs, but they're interactive and dynamic protocols. So there's a lot of stuff happening here, which I will show you in a second. And so all public protocols on Protocols.io are open access. That means everybody can access it without even needing to log into anything. So it's all free to access. There's about 4,000 public protocols right now on Protocols.io. And it's not only molecular biology protocols, but also different fields such as psychology, a lot of computational workflows, and all different kind of things. And so this is what you see here is like what usually the main page of a protocol looks like when you click on a protocol. And so one thing here you see, it can be associated with a group. This is an example from a human cell atlas project here in a protocol. And then also here you see the step-by-step description. So this is only a first step you see, but there's single steps, so it's not one big blob of text, but it's like separated steps, so it's easy to follow when somebody's following this method in the lab. Another thing is here on the right-hand side, you see a commenting functionality. So this is really to discuss the protocol, and this happens on a private level, but also on, like this is a public protocol, so this happens on a public level too. And this is really to discuss the protocol on two different levels. And one is called a protocol level, which means it's a discussion on the protocol as a whole. And then there's also step level discussion, which means it's a discussion on a specific step. So if, for example, you find a protocol and it says in step 3, 30-minute incubation, but you think, oh, should be longer, you can leave a comment and ask them why they did 30 minutes or suggest something different. And one thing to note, I want to say here, because if you're using this discussion on a private level, if you're ready to publish the protocol, you have the option to remove the private comments. So keep that in mind. You don't have to publish your private comments with it. And of course, every protocol can be associated with a publication. So not only the publication has a link to the protocol in the materials and methods section, but also the protocols link back to the publication. And then every public protocol gets a DOI, a digital object identifier. That's really to keep the protocol it's permanent, and this link will always link to that specific version to the protocol of the protocol. And then another thing, the next thing is we have something called versioning. So that means if you publish a method, once it's public, and you cannot make any more changes to it. But the beauty of protocols.io is really that you can keep your method up to date with something we call versioning. So at any time, if you need to make a correction or any improvements to the method, you can always create a new version of that specific method. And then the viewer also has the option to compare between different versions, so they can see what changed between version 1 and version 2, or if there's version 10 and version 15, you can always put them side to side next to each other and see what changed. There's like a little thing that highlights the changes, so it's really easy to see what improved or what changed between the different versions. And also, if somebody, for example, has the DOI for version 1 and they click on it, they will get a little notification that says that there's a new version of this protocol available, and they can decide which one they want to see. And then we have something called run, but I'll show you that in a little bit. It's really to have a checklist functionality that you can use to follow along while you're doing the experiment in the lab, and you can check off every step, and you can add notes to steps as you follow the methods. And of course, if you want to print it out or have just the protocol as a record, as a PDF or wherever, you can export it directly to your computer or Dropbox, Google Drive, or Box. So you have that option too. And when you create, when you create a new protocol, first, when you create the protocol, it starts off private, so it's only visible to you. It always starts out private, and then it's really up to you who you want to share it with later. So you can share it privately with individuals, or you can share it with groups, and there's different access levels too. So if you share it with one person, you can, for example, say, do you want that person to be able to edit this protocol, or should they be able to edit and publish, or maybe you only want them to be able to view it, and so you can do that. And then it's up to you when you publish it as well. So it can be private for however long you need it to be private, and then you decide when it goes public. And so this is just like a little animation here of our editor. So there's a lot of tools you can work with to just add a lot of details into your step. So you can add images and timers and videos and really anything that could be helpful. But this will also cover more in the second part. What kind of details are useful for this? And so here's another close-up. You can group the steps into different sections. So if there's like one section that contains multiple steps, you can group them, color code them, add timers, and add like really a lot of detail here. And here on the right-hand side, you see all the components that you can use to work with. And then here you can add the videos. And then also, as I mentioned before, it's not only wet lab protocols, but you can also add computational parts to a protocol or even entire computational workflows. So you can include all your data set, and you can put your code and link to whatever you need to link to in your computational workflow. And once you're ready to publish your protocol, there's really two different options. One is called Get DOI and one's called Publish. And just to keep in mind, when you're ready to publish your protocol, the difference between those two is that if you click Get DOI, your protocol will get a DOI and it will be permanent. You can no longer edit it, but it will be still kind of hidden because it will not be discoverable through our search functionality. Or if somebody searches for it on Google, they will not be able to find it. So this is really if you're submitting a paper and you want to get a DOI that you include in your manuscript for the reviewers, then you can use that option because everybody who has the DOI will be able to see it, but anybody who does not have the DOI will not be able to find it. And if you're ready to make it fully public, you can click Publish, and this will also give your protocol a DOI, and it will be permanent, so you can not make any more changes, but your protocol will also be discoverable. So if somebody comes to protocols.io and they search for your protocol, they will be able to find it. So those are the two differences to keep in mind. And of course, once it's public, it's citable. People can cite it, you can add it to your manuscript, and also every public protocol, we archive it in clocks to ensure long-term preservation. And of course, once it's public, people can comment on it. They can create their own version of your protocol called a fork, so they can create their own copy of your protocol. And of course, you can always keep it up to date with the versioning that I explained before. And one functionality that we have on Protocols.io is running protocols. So this can either be done on the web or mobile. So we have free Android and iOS apps that you can just download, and you can have all your protocols on there, and you can basically check off all the steps as you follow the method along in the lab. And then you have the option to add notes to each step or make changes if there's any changes that you made on that specific day. And then of course, every step as you check it off gets a timestamp. So later when you export them, the experiment record, you will be able to see when exactly that step got checked off and when the note was added and things like that. And now I'll hand it over to Lenny again to talk about the current status of Protocols.io and where we are right now at.
And thank you, Anita. Can everybody see my screen? Am I doing the screen sharing?
Yes, we can. Excellent.
So one of my favorite examples of where we are now, Anita mentioned that there are over 4,000 public protocols, and I'll talk a little bit about where we are, how quickly it's growing, how long it took to get here in just 1 or 2 slides. But even already, even though it's still fairly early, we got started in 2014, it took time to ramp up and get recognition and trust from scientists and get scientists to start sharing. But already with the 4,000 public protocols, we're at a point when things like this are possible. And so this is one of my favorite examples. Here is a researcher from Chile who is asking on Twitter, does anybody have experience doing RNA extraction from cortical neuron cultures? And a postdoc from UCSF, Elena, she replies saying, you should try this particular method called RNA trisol extraction. And then she says, I see on Protocols.io, there's several, and from those that I see on Protocols.io, here's one that should work. And what I absolutely love about this example, why I'm bringing it up, is not just that she found one of the recipes that works, that should work for this researcher, but if you look at that protocol, it turns out that it accompanies a published paper in the journal GigaScience, and that paper is on parasites of stickleback fish. So the RNA extraction protocol that accompanies this paper has nothing to do with primary cortical neuron cultures. It's for the parasite of stickleback fish. And so what I love about it is that not only does including a link to this protocol in the paper improve the rigor and reproducibility of the paper itself as it's published from this group, but by putting it in the central repository, you empower other people to take it, as Anita said, to create forks of it, to use it in their own research, sort of use it off the shelf. So it goes far beyond just the utility of the people reading your specific paper, because now it's in a common knowledge base, and other people can take it, adopt it to their work. And so there is a real amplification that happens when you're sharing in a common resource. And where we are right now in terms of adoption, it took a while. As I said, we launched in 2014. In the beginning, as Alava heard me say, no one knew that we existed, and there was a period of number of years where it was barely moving. But as you can see now, we're finally reaching that part of the nice part of the curve in terms of adoption. So more and more people are sharing protocols, more and more people are coming to Protocols.io to discover this knowledge. And at this point, as I quickly mentioned, we have about 4,500 public protocols, including versions, 4,000 without them, 15,000 private protocols, and it's growing at just over 1,000 new protocols per month with 800 private, 150 public. A lot of people, as they submit protocols, they're doing it as part of papers that they're trying to publish. So there is often a lag, a delay where protocol starts out privately and then moves into the public space once the paper is published. And speaking of the publishing, just 2 years ago, there were only 3 journals that had Protocols.io in their author guidelines, encouraging scientists when they submit papers to detail the protocols and Protocols.io and link to the DOI that Anita showed from their paper. So 2 years ago, there were just 3 such journals. Then Public Library of Science in 2017 in spring added us to all of their journals. And after that, it grew really quickly. And so now there are over 500 biomedical journals and those outside in geosciences and psychology. So it's starting to branch out beyond our roots in life sciences. But now there are over 500 journals that have Protocols.io in author guidelines. And with that, I'd like to acknowledge an extraordinary team that's working daily to make Protocols.io better. So that's why we're looking forward to the Q&A, and that's why we're always looking forward to feedback from you as you play around with Protocols.io, as you start using it. Click the help button. You have my email, Anita's email. Never hesitate to email us. We have a weekly brainstorming meeting where we go through all of the feedback we got over the previous week and prioritize what we're developing to make sure that we're building a platform for the users and not for ourselves. And I would also like to acknowledge our generous funders, which is the Gordon and Betty Moore Foundation, the Chan Zuckerberg Initiative, and the Open Philanthropy Project, and our earliest publishing partners, which is Genetic Society of America, GigaScience, and the Public Library of Science. And with that, I will stop the screen sharing, and Anita and I will be delighted to answer any questions.
Okay, so this is our break at the halftime break, say. So they'll talk some more later, but we open up for the first round of questions. We can either take written questions. I don't think we have any at the moment. You can also raise your hand, and Juliana will put you online. You can do your questions by live as well. I mean, I can start if nobody else. Really, I mean, to us, one of the things is like, how can we make this more useful in terms of like the format we already have? What's the best way of sharing? So, I mean, just to remind you on more or less what the initiative workflow is, we start out with literature protocol, and then which is of course incomplete. I mean, I talked about it. There's like lots missing always, or mostly. Sometimes it's like very, very brief. For a whole experiment, which means it goes from like injecting a mouse with something, then dissecting the liver, then doing RNA extraction, and then they end up doing an RT-PCR, for example. And that actually becomes 3 slightly different protocols, because like each one of the teams who's replicating is independently filling in the gaps using some standardized forms which we made ourselves. So like we end up with like 3 slightly different versions of each experiment, which really is, I mean, it can be a lot of steps, right? I mean, it could have like an in vivo step and an in vitro thing. So, I mean, so my question is like, as you see it, what's the best way of actually like contributing with that? I mean, should we have like a single protocol for every experiment with 3 different versions? Or like, should each version be a separate protocol? Or do you actually think that for an experiment it would be better to like break it up in like 2 or 3 protocols in terms of like being more useful, like just have the RNA extraction in one part and the whatever in vivo pharmacological injection in another? So how do you visualize organizing this all within protocols to be like more useful to the community?
So I think there's a few questions in here. One question, the simpler one, is if you have a very complicated protocol, do you share it all with 300 steps, or do you break it up? And we have examples of both. I think, Anita, you probably know better what our longest protocol is, but I think we have some that are 200 steps, if not more. But if it can be broken into discrete parts, like tissue preparation, you know, you're dealing with lung tissue. So one part is lung tissue preparation and dissociation into single cells, for example. The next part is the sequencing part. So what we recommend is if you can break it up into discrete tasks, then maybe make it slightly shorter protocols that contain individual 1 day or 2 or 3 day experiment, which is concrete, and other people could then take it and plug it into their own pipelines, right? And then so you would share lung protocol, you'd share the tissue dissociation, the DNA extraction, and then the DNA sequencing and analysis, for example. So those could be 3 protocols, and then you just add them into a collection. So when you click plus on protocols.io, we just didn't have time to go into all of the details, but you can combine and shuffle around different modules, different protocols into a collection that gets its own DOI. So very often when people cite protocols from a paper, instead of citing 3 or 10 different DOIs, they put them together into a collection, and they will say the media, buffers, and culturing conditions for our Drosophila experiments are at the following DOI in a collection in protocols.io. And so then all 10 protocols could be in that container. So that's the answer, that's the recommended answer for how to split up protocols. You don't have to, you have the flexibility, but then other people can take them and create their own collections and put in maybe just the sequencing part, but they're doing extraction differently because it's a different organism or different tissue that they're dealing with, right? And then they don't have to make a copy of your protocol and go through and delete the first 100 steps and use just the last 100. So that's the easier answer. That's collections. The second part of your question was, what do you do if you have sort of one master protocol and then everybody else is branching off and has their own versions of it? And
yeah, and actually, just to make it more complicated, like the master protocol is actually from somebody else's paper. So I mean, I'm not sure you can you link it to somebody else's article?
Yeah,
because I mean,
there is a link. Yes. So I actually just did a webinar 2 weeks ago called Copyright, Credit, and Scooping, where we talked about, can you take somebody else's protocol that was published in a paper and put it into protocols.io? What about copyright? What about plagiarism? Do you need to change words? So that's a whole separate half-hour webinar. But the quick answer is no one has ever complained. No publisher has ever sent us a takedown notice. And in the United States, at least, methods are not actually copyrightable. So the United States Copyright Office says that instructions and methods and recipes are not copyrightable. They're statements of fact. So you can't take a paper that's copyrighted and put it in a different platform necessarily, unless it's Creative Commons. But as long as you cite it, as long as you say this is coming from that paper, if you put it into protocols.io, you should be fine legally. And we've never had a takedown notice from a publisher since we got started in 2014. But, oh, and there, Anita just shared the link to the copyright webinar. But I think one of the hardest questions that you asked is, if you have existing protocols, what do you do, right? And there are different flavors because you're sending it out to 5 or 10 different labs, and they have slight variations of this. So we very much want to capture this information. This is at the heart of protocols.io, why we have the copy, fork, and versioning, so that we're not a journal that's trying to share one protocol, the master protocol that everybody uses. We are trying to capture the space of variations around protocols so that people know in Grasshopper, this was the protocol that was used, but if you're doing research on ladybugs or bees and you're getting RNA out of them, there's slight tweaks. We want all of those. So my answer would be, for your initiative, maybe send the master protocol into protocols.io, and then the versions could be sent in as attachments. You just attach them to the file, and you say in the abstract, below are variants on this from 5 different labs. And then you can always create a version of it and share forks off of it. If one of those becomes really popular and everybody needs it, you can take it out of PDF, and we can we can either work with you and help you get all of those variants in. But maybe the simplest way to do it is coordinate with us on pulling in, when you're ready, the master protocol that you've given to others, which may or may not be from some published paper. right? But you've assembled the master protocol. And then as the other labs start publishing and they have finalized their tweaks and their variants, those can be attached to the master protocol so that you capture all of that space.
That's different versions. Because I think that
and you can create a new version of the master protocol, adding more and more attachments to it, saying here are the other 10.
Yeah, but I like the idea of being able to compare, right? I mean, for us, actually, like...
So to be able to compare, we would need to, if they're similar, if they're following a standard, we can work with you to actually import all of them, keep the relation between them, and then give people what Anita was showing, that ability to click a button and compare them side by side to see what's the same and what's different on the individual steps. So that's doable, but obviously it takes a little bit more work because you already are using Google Forms and Excel and whatever format you're using. But it,
we'll end up with like a written protocol, which I guess that's another question, like how do we submit it? I saw that you have some offer of like submit whatever and we'll do it. I mean, but we have like, I don't know, like 180 protocols. So I mean, I'm not sure if you can do it or we should do it. Like maybe that's a conversation to have afterwards.
Well, we can just, I think there's many participants. We can discuss online until the end of July. Any user can actually just send us, if you have a complicated protocol, you can send it to us and we'll put it in for you. So that's something that Anita and us are running the entire month of July. So if you have something that's been sitting on your desk, because our editor is good, it's easy to use. If you have a simple protocol, a buffer recipe with 10 ingredients, it'll take you 5-10 minutes to put it in. But if you have a complicated protocol with 200 steps, that might take you an hour. So we're running this offer where you can send, any user can send up to one protocol to us and we'll get it in. But of course, if you have 180, hopefully there's some standard to them and we can parse them, but then, you know, we need to connect offline and just see what's the best way of doing it. There's also a question, which says, hello, I'm Clarissa, a postdoc in immunology. My question, which are the main obstacles for people to share a protocol? Is language an obstacle? In France, several members of labs around me would resist to write in English. It's a really good question. We don't do translations. We can't localize the 1,000 protocols that are coming in every month. So language is a barrier. When Anita is going to talk in the second half about the art of sharing protocols, that's actually one big concern. As Americans or Europeans from Britain share in English, how many people can understand it, whatever language you choose, right? And we know at Protocols.io that only 40% of our traffic comes from the United States. It's very global. There are people from China. There are people from Argentina and Brazil, right? All over the world. And so that is an important question. We have people who have been sharing protocols in Spanish. We've seen that. Obviously, that then suffers from the flip of the problem. English is for English speaking. If it's shared in Spanish, that is accessible to those who are Spanish speaking. So there's no great way around it. If you want to share it in Portuguese or in French, you can do it. My recommendation would be to make simple videos, 10-second videos. You go into the lab. If they're complicated, and this is something we advise to English speakers who often forget that there are people around the world who don't necessarily know English. If you have complicated steps, make a video of it, put it right into the step, and that's a natural localizer. That's a natural translator that even if your English is not perfect, now you see how people are doing it. So one recommendation is think about videos. But that's a hard question. There's another question. If somebody can translate, oh, I think that was someone talking about video.
No, that was just video. Yeah. But there is a second question though on pre-registration of methods and analysis plans. I mean, do you is that meant for that as well, or?
Oh, analysis plan and pre-registration.
Yeah, pre-registration.
We welcome. So we are very open. So the question, the other question is, you know, is it just for sharing methods or can you pre-register? We're seeing actually more and more study designs coming in, including with human trials, where people are describing this is what we will be doing before the work starts. So it is being used as pre-registration. It gets fixed. there is a DOI, we welcome that. So describing the analysis plan, this is a great use of Protocols.io, but if there is a better platform or the journal itself has a pre-registration forum, of course, you need to choose for your field, for your community, what's the most appropriate place. Some of that can go on a OSF, some of that can go directly to the journal, but we welcome pre-registration, and we are starting to see more and more of those. Another question.
It's the same question, Lenny.
Oh, okay. I see that. Excellent. So we love the questions. And by the way, as you keep, if we don't get to all of them, keep typing in the questions. Anita and I can then, if we run out of time, we can write up responses, and then maybe Juliana and Alava can send it out to participants or put it up on the page. So keep asking, but I think we should transition to the second half, and then we'll have more time for Q&A as well. So we'll try, we would love to get to as many questions as we can. These are excellent questions you're asking.
Cool. But yeah, so I guess you can move on and do the second half, and then we have a new round of questions at the end.
All right, so I should be sharing now. And as I said, as I said in the beginning, this part is less about the platform itself. You can share on Google Docs, you can share on GitHub, you can share on Figshare. There are many public places where you can share a protocol. And I think one of the key important questions to keep in mind is wherever you're sharing, Protocols.io or not, what goes into writing a good protocol? Part of it is language, as just came up in the Q&A, but part of it is what we actually run a workshop called The Art of Writing Methods. We won't be able to do the full workshop. It's an hour, it goes in person, so it's hard to do on the web, but I can describe what we do. It's one of my all-time favorite lessons and hands-on activities with Protocols.io. So we learned this from a professor. We learned this from a professor right there. From a professor on Twitter who said, I just did this exercise with my students in the class where I had the undergraduates draw something. That's what you see on the first image. Then you write the instructions for it and give it to somebody else. And you exchange notes, and you have to reproduce what was done. I have the link here. We'll share the link to this workshop. So Alava and Juliana, feel free to share the slides. So we took this exercise, and you can see the house and the person next to it. You can see the instructions for it and a person trying to reproduce. And instead of the house with a chimney, they got a house with a huge, enormous, giant brick on top, right? And that is just a beautiful illustration of the art of writing methods. We've done the same at many workshops where we have people exchange, draw something, write the instructions for it, and then swap with another person. Sometimes it works out brilliantly. Here you can see, is this an experiment that reproduced or not? It's very close. The instructions weren't terrible, but instead of a frog, you sort of have a cartoon person, right? It's not Xenopus. Here, the instructions, I've actually looked in them. In this example, the original is on the left. It was a house. And the instructions weren't bad, but the person who was replicating it, for some reason, thought that this is a rocket. And once they thought that it's a rocket, they followed the instructions and turned it from a house into what they thought prematurely it is. And then another example, I won't go through too many of them, but another example is this very simple drawing, but making instructions for it is a nightmare. It's really hard. The person who got the instructions struggled for 20 minutes and then just gave up. And this is what we have on the right-hand side. And so there are a few lessons that you can learn from this, which is you will never get a protocol description perfect in the first place. It takes time. You need to see what do people not understand. They will come to you with questions. They will make mistakes as they run it, particularly if they come to your lab to train with you. And so getting the feedback, kind of like having your paper peer reviewed, there's a natural peer review of protocols. This is why we encourage commenting and discussions on protocols.io, and we see a lot of versioning after discussions as people try to make their protocols clearer. Where did you order the chemicals? What concentration did you use? What do you mean by grind the tissues? So those questions, as they come in, right, as you see what people are getting confused by, you improve your instructions. And when we don't have enough time to have people swab, this is an exercise, a variant of this exercise that Anita came up with. We have a 5-minute version of this where we tell participants in a workshop, here are instructions. You don't know what they're for. Please follow them and try to reproduce the original. And this is something that you could go through on your own, but we'll give you the answer. What happens when we let people run through this exercise is that from the same protocol, you get quite different results, right? So these are just examples of different people. We've run it many times now, and it's always fun to see. These are different people following the same set of instructions. And what this tells us is that this set of instructions can be made much more precise. As we look at what did people get wrong, we can go to that step and write a better version of it so that everyone would actually get very close, right? So there is an art to writing a good protocol, and it needs versioning and needs iteration. So this was the original. It's from a cartoon. It's SpongeBob, and the same set of instructions. Some people get very close. Some people, you wouldn't know what we started with. And that's one of the things that I think Anita will highlight next is how do you get to this point of sharing good protocols, and how do you choose the platform where to do it, and what are the important aspects. So I did just say, I think I said exactly this, that it's not just the platform where you're sharing, but how you're sharing that is very important. And so with that, I'll give it over to Anita.
I think I was muted. Okay, so I'm just gonna walk you through some best practices for writing reproducible methods now, and show you also some good examples of what that could look like for your methods. And so one thing, I think Lenny mentioned it before, it's really important that the methods are digital. And so because if you have a protocol and it's either in your paper lab notebook, or maybe even just on a piece of paper somewhere, or you just have it printed out, but you don't actually have to file to it, there's a lot of issues with that. One big issue is it's really hard to share that. It's not searchable. If you're trying to search through a paper lab notebook, if you ever try to do that, you might know that it's very difficult. And also it's really hard to make duplicates of it. So I really, really encourage you to have a digital version of it. You can either use protocols.io, but you can also use different platforms, like have a Google Doc that's easily shareable, or any other electronic lab notebook, just to have it easily accessible, and you can share whenever you need to, or you can search through it whenever you need to. And one thing for protocols, we like to think of protocols as a brief, modular, and self-contained scientific publication. So see your protocol not as just part of your publication, but see it as its own little publication. And so usually a publication has an abstract that just explains what the publication is about. It's really, really good to include an abstract for a protocol as well, because the protocol might be sitting somewhere for a couple of years, and then somebody might pick it up and they have no idea what you originally used this protocol for or what it was all about. So it's really good to put a little abstract that just puts your protocol into context and just gives the reader a quick introduction to the method so they know what it's all about. And then, of course, it's really important to include as much detail as possible. A lot of times I hear people say, oh, this is everybody knows that, it's common knowledge, they don't need to put they don't need to specify different details, but it's really important to put a lot of detail. It's always better to put too much detail. Actually, I don't think you can put too much detail, but if you just assume that everybody will know, I think that's not going to go well, because you should assume that people don't know the details. And so you can put exact duration. So if you do an incubation, put exactly the time, exactly how much of reagent amount did you put in where, and also the reagents, which vendor did you order the reagent from, which catalog number did the reagent have, maybe even the lot number, because sometimes lot numbers even matter and can make a experiment not be reproducible. And just like include as much detail as possible. Also, for example, if you have any images of what the result might look like, or videos or whatever, can help to make the protocol be more understandable. And that's also related to the language. So if you have images, like pictures talk more than language, and they're internationally, so it's really good to add images to explain also what your text is saying, so other people will be able to understand it easier. And then also one thing, in scientific publications, we tend to mix up the chronological order of steps. So try to keep the steps in chronological order. Just start with whatever you would start when you're in the lab doing the experiment, and then go on however the steps are as you follow. So don't say stuff like, oh, prior to this I did this, and then like after this, like just keep it in order to make it easy for everybody. And then also try to limit jargon and use active voice. So if you're writing the recipe, really try to put active voice. So don't say what has been done, tell user what they should be doing if they're following this method. It just makes it much easier for somebody to follow that method. And of course, add as many notes and tips and tricks to the protocol that might be helpful for somebody who's seeing this protocol for the very first time and have never followed it before. And here's some examples of what a good method detail would look like. So here, for example, this is a very popular method on protocols.io. It has a brief abstract that just puts the protocol in context, and everybody who uses this protocol or comes to protocol can read the abstract in just like 2 minutes and know like what the context of it is. And here is a protocol example that's in the running functionality. They included the timer, so that just makes it very good, because you know exactly what kind of time you need to air dry the beads. So it's just not air dry beads, you know exactly the time. And another thing, I mentioned it before, is really good to include images and videos. Here on the left, there's it's one of our users in Africa, and she's a soil scientist, and she includes these really beautiful images to just explain how to do things. Like, for example, here she shows how to cut the tip of the end of a plant, and she had trouble finding how exactly to cut this. And so when she published this protocol, she said, well, I'm going to show people exactly how to cut this, so they don't have to wonder like which angle it should be or whatever. So it's really like this, creating something like this, it just helps a lot to make your protocol be more reproducible, because people will see examples of what you also put in the text. And then on the right hand is a great example of one of our users. He updated his protocol with a new version because first the step said to grind a tissue something, and then he thought, well, this grinding might be confusing to people and they don't know how exactly to do it. So 2 people in the lab, one person was doing the step and another person was just filming it with the phone, and then they uploaded this to the step, and it just adds so much value, because people who are now following this method will be able to watch this short clip and they will know exactly how to do it. So it's just really helpful. And another thing, I mentioned it before, what's really important is the reagent, because a lot of reagents like this can really be the factor that makes your method be reproducible or not. If you order it from a different vendor, they might have like different compositions and something might be different, and then your method might not work. So it's really important to put very detailed reagent lists. Here's a good example where they included all the materials, and then they included catalog numbers and the vendor, so you know exactly what reagent is from. So that's really good. And Also, another thing is the equipment is really important. So, like, for example, if somebody says shake for 5 minutes, like there's so many different kind of shakers you can use, because one shaker might rotate, one other shaker shakes like a different way. So it's really good to also specify which equipment you use for a specific step, because not all equipments are the same and it can make a big difference. And here I just put a little checklist that you can look at when you're publishing a protocol or when you're creating a method, to just look at to make sure it's as reproducible as possible. So is it digital? Did you include your abstract? What's the status of the protocol? I think Lenny was mentioning this before, like, is the protocol something that's working? Did you try it? Maybe it's something that's in development. Maybe also you tried it and it didn't work, so you can also publish a method that didn't work for you. And then also the details like images, reagent details, equipment detail, timers, etc. And then the active voice again, and make sure your steps are in a chronological order, and you can make all your data available if you have any data to support this method, and any additional tips for that method. And then I'll hand it back over to Lenny.
Thank you, Anita. And because we started this part just a little bit late, and I want to make sure that we have time for question answers, I'll do just 1 more minute on the next part, and then we'll just go right into Q&A. But if you want to hear more detailed take on this discussion of when to share, I added back again the link that Anita shared earlier. It's in the chat. It's the webinar that we did recently on copyright sharing, and we go through this part explicitly. So take a look at the chat. And the part that I was going to mention just briefly is the question, we've talked about sharing as part of publishing papers, and just now we're talking about the art of writing a good detailed reproducible protocol. But one thing we didn't touch on is, when do you share publicly? And one of the things that we think about a lot of Protocols.io that we strive and try to encourage people to do is to share before you're ready to publish, right? Because as we develop methods, as we troubleshoot and iterate and try and fail and optimize things, there's just so much knowledge about what we've tried to get DNA transformation to work in a particular marine single cell exotic creature. There are things that maybe didn't work. And it would be really useful for people to know what you've tried. There may be things that are working for you. So in addition to the negative results, we've tried the following things for the last year. You may never publish that because it didn't work, but sharing these things before there is a paper, and maybe there will never be a paper because these are things that failed, is important. It is also important when things are working, even if you haven't reproduced it maybe 1 million times. If other people are struggling with the same thing, sharing the protocol, it's not a prior publication. It won't, you're not scooping yourself. You'll still be able to publish the paper. It's not even a preprint yet. It's just the protocol part. But there's this question of, can I share early? And some people hesitate to share prematurely. So what I'll say just in 1 minute is, what we've done at Protocols.io is, one of the parts of the workflow for publishing is we require you to set the status of your protocol. So as you click publish, you enter the authors, and then we ask you, can you please self-certify? Is it a robust working protocol? Is it something you're still developing and maybe it's tentative? Or maybe it's a negative result, right? And you have to pick one of these before you can proceed. So if you are sharing early, if you're not sure, you can just say, we're not sure about this protocol. And then when it's published, and you can put in a note on what that means. And then when it's published, you can actually see there is a status of protocols. There is a status of protocols, whether they're working, as in this example, or whether it's something else that was attempted but doesn't necessarily work. And so I went through that a little bit quickly, but I think it's best if we stop here and see if there are more questions.
So we're open for questions again. We have one here. So again from Clarissa.
And the question is, do you have strategies to reach more people, spread the word, eventually ramifications of the platform in other continents? And I'm smiling because that is, Olava has heard me give a talk on this at the conference where he and I met. That is probably one of the hardest things about launching a platform like this. It's not even raising the money, which is hard, or building a useful tool. That's also hard, that's engineering. But reaching people so that they know that this exists is one of the most challenging aspects of getting an initiative like this off the ground. So we've done many things. We've gone around different companies and labs to get popular methods that people trust. So first you need content, and that will bring people from Google. We know that people, when they come at first to Protocols.io, the first thing they do is not put in a protocol and share. The first thing that they do is look for a method that they use. So even if you get people, if you spread the word and you get people to the platform, you actually need content that they're looking for. Because if they don't find the methods that they use, they say, oh, this is too early, I'll come back in a year. It's a nice initiative. So it is a hard thing to do. We've never advertised on Facebook, Twitter. We don't think that that's wise use of the budget, but we do try many things. Like, this is one of the reasons why we go to all the publishers, because when they add us to author guidelines, that increases visibility. And every time a paper is published that has a link to Protocols.io, that leads to many people discovering it. So there's a lot of work to do. I think we had 500,000 scientists visit the platform over the past year. There's about 100,000 views of the protocols, of the public protocols every month. But that's still just a fraction, 10, 15% of all the scientists worldwide. So yes, there's a lot more to do. Anita runs an ambassador program across 5 different continents. We have students and postdocs who advise us, who help us to spread the word, who give us feedback on the platform. We have monthly calls with them. So if anybody wants to help us with the feedback and wants to join the volunteer ambassador program, get in touch with Anita. But it's a fantastic question. It's not easy to do. We do a lot, and we probably should be doing more to spread the word and get the content and get adoption and get the trust. It all takes time. Fantastic question. Thank you, Clarissa.
Yeah. Well, I don't think we have any. I have a couple more questions. One simple one is, can you actually associate a result with it? I mean, just include sample data within a protocol? Because, I mean, it will be very, like, we have this huge publication, like 60 replications and like 3 versions of each. I mean, a very easy way to see which protocol corresponds to our results is just like having the result with this. So, I mean, can you do it? Can you just like put in a figure or something?
Anita, do you want to answer that?
Sorry, I was going to say, I think somebody else has a question. I think they raised their hand.
Oh.
And you were cutting out a little bit. Can you repeat the question?
My question?
Yeah. I can answer then, Anita. Yeah, so let's see who raised the hand. Yeah, I do see one raised hand, so let's make sure we get that person's question. But the question from Olava was, can you share results? We actually have on each protocol a section for results. And we also often see people sharing just in the comment section. They will put in an image. You can always create a new version. You can attach files. But we have a dedicated results section where we try to get people to share, this is what the quality of the data should be. This is the distribution of your reads if you're doing DNA sequencing. So we do love it when people share what the protocol produces. And most of the time we see the results shared when people have questions. So they will ask a question to the author saying, is this what I should be seeing? And they will attach an image of what the protocol produces for them. Fantastic question. And let's see, Pedro, you had a question? I think you're on mute. You'll need to unmute yourself.
My question is like, methods, when you get like when they're written in extremely exhaustingly detailed, they tend to get really long, and they might kind of overburden reading if you don't actually need to read every single detail. So you do think every method should have like 2 versions, a brief description, and you kind of outsource another one with all the information. And how, I mean, the brief one is kind of what we get in published papers, because the journals don't give a lot of space. But I mean, I don't know what should we do in terms of guidelines for publishing methods.
Anita, you should take that. You just went through that part.
So, so actually, I've seen that some of the protocols have 2 versions. Like one is like a detailed version, and one is like just more keywords instead of like entire sentences, for example. But also in Protocols.io, we do also have an option to add guidelines. So everything, for example, that is not necessarily the step, so something that's not actually the action that you have to do, we put into guidelines. So you can have like a longer guideline section that just describes things, but then the steps should be kept fairly short. So you should, so you can make those different points. But also, I guess if you have a long version of a protocol and somebody knows like most of the things, they can always create also their own fork of it and kind of make it shorter and cut out whatever they think like is unnecessary for them as they're following the protocol in the lab. So I would say like, yeah, if you want to put 2, you can put 2, but if you only are going to put 1, put the more detailed one. So yeah, it's up to you, but it's, I guess, it's, if you have the time to put both, you can put both, like a long version and a short version, but I think the longer version is always better, so you can have more people understand it, and then they can always cut out whatever they think is excessive and they don't need it for themselves. Does that answer the question?
Yeah, it was exactly about that. But I was actually talking more about like the publication system itself. So like how that should be, how do you guys think about how it should be on journals, like not necessarily on the platform? I don't know.
We've had many discussions with editors and journals about it. So my personal view is, it's not that we don't need materials and methods section anymore. So if the question is, how do you balance the details on the protocol versus what should be in the paper? Not everybody's going to go from the paper onto Protocols.io and follow the link. Not everybody needs the full 10 protocols in your collection with the details of the media and how you create it. That's only the people who are going to follow up on it. So in our view, the materials and methods section is kind of like an abstract for the method section, right? So the method section should have the abstract that quickly lets a person reading the paper know, oh, this is what they did. They used the appropriate technique for the research and for the results that they're producing, so that at a glance, kind of like the abstract for the paper itself, at a glance you can tell, what did they do? Did they use the right software? Did they sample correctly, right? Did they go, which ocean did they get this from? Will I agree with their results? And then for the people that need the excruciating detail, that's what goes into Protocols.io. So it would be a disservice, even if you remove space constraints. It's not that journals are terrible. Even if you remove all space constraints, if you now make your published paper 58 pages with 50 of them being protocols, it becomes very hard for the reader who's not following up on it. So I think our recommendation would be that journals maybe require a little bit less. Don't try to fit all of the protocol condensed into 2 paragraphs. Put it in appropriately into Protocols.io, link to the full details, but still have a short description in the materials and methods of what was done. But great question as well.
Okay, do we have more questions? I have a final one. I think you had it before. I mean, you mentioned it in the beginning and suggested it, but nobody asked yet. I mean, what's your data preservation plan? I mean, what happens if the platform goes broken? I mean, does it all go away? Do you have like a set fund for that?
So the question is, what happens if we go bankrupt? What's the long-term preservation plan? We have many. We answer it on the FAQ on our /help page explicitly because it's a very important question. It is frequently asked, so it's in our FAQ. But quickly, as Anita mentioned, every public protocol we send to the digital library archive called Clocks that all biomedical publishers do. So if anything happens to us and protocols.io in 50 years it no longer exists, the PDFs will still show up. The DOIs will resolve from published papers. So for public content, we do archive all of it in Clocks. It's a nonprofit from Stanford, Digital Library Archive. We also mirror everything in a GitHub repository, searchable GitHub repository. There is a link from our FAQ. So if something happens to our servers, and Anita just added the link to the FAQ, if something happens to our servers or Amazon and you can't get to the method that you need, you can always go on GitHub. There is a PDF and a JSON format version of each protocol, public protocol that's available. For the private ones, of course, we do backups, but we don't put them on GitHub because they're yours and they're private. But you should always, whatever lab notebook, electronic lab notebook, whatever platform, whatever tool you use, you should have ability to easily back up everything. So we give you one-click ability to download your entire folder with all of your protocols as PDFs to your computer, one click to send it to Dropbox or Google Drive. Microsoft OneDrive is coming. So we encourage that. And then we also have public APIs so that people can plug in and pull out protocols as well for the public and private. So it's an important question. Nobody can ever guarantee 1,000 years of preservation. It's a very good question, and those are some of the things that we've done so far.
Okay, do you have anybody else? Not on the chat. If there's no more questions, we can wait a minute or two. But I guess I should thank you for showing up and talking to us. We will have a couple of questions, but they're probably longer to solve, and we'll do it in time. Currently, just to let you know, we have our protocols are currently kind of like a form-like structure. We're asking whatever's missing in a form structure. So we have to eventually see when do we translate that into text, and then probably send it to protocols.io. But this is something we should talk about internally. Actually,
we'll do another call as you move forward with your reproducibility initiative. We should do a call and just have a good plan so that we're not then scrambling and doing too much additional work. I think there should be ways to optimize.
Yeah,
that.
And second one is the one I was talking to Anita. Just can we have a big group with the separate privacy for each document? Because we want people to actually develop protocols independently. So she said she was working on that, but we can discuss that later. But it's just about how do we structure our own group in the best way possible. And
so we're happy to do a call and discuss that. But also for anybody else who might have questions tomorrow, later, or if you're watching this as a recording and you have questions, our emails, I just added to the chat, our emails are Anita@protocols.io and Lenny@protocols.io. So feel free to email us or use the contact form on protocols.io. We'd be happy to answer any other questions. Yeah. And thank you, Juliana, and thank you, Olava, for the invitation.
Yes, and this will also be made available. It's probably available already in our Facebook page. We also, if you have nothing against it, we should also post it on our.
We would love that.
Our YouTube page, just so people who couldn't show up today but want to watch can do it later. So thanks a lot, and thanks Lenny, thanks Anita. I'll open our team to questions now. I'm here. Pedro's here. Kleber is also here in the room somewhere. So if people from the initiative want to talk about how the protocol development thing has been going, you're happy to make your questions to us as well. So thanks, Lenny. Thanks, Anita.
Thank you all. So we'll unplug with Anita, and then you can have an honest discussion without us auditing and eavesdropping. Yeah, thank you all. This was a lot of fun. Thank you for the fantastic questions.
Wow. Thank you. Bye. Bom, gente, a gente mandou os protocolos semana passada, tem pouca gente aqui ainda, deve ter 4 ou 5 pessoas, mas enfim, tem colaboradores nossos. Se alguém tiver alguma crítica ou algum comentário sobre como é que foi o processo até agora, acho que talvez já tenham mandado os protocolos, estamos aqui. Se não, vocês podem fazer isso por e-mail, vocês têm o nosso e-mail, etc. E se ninguém entrar...
Parei lá no Facebook um pouco antes e agora estou parando a gravação aqui.